Cellulosic ethanol is a biofuel produced from wood, grasses, or the inedible parts of plants. It is a type of biofuel produced from lignocellulose, a structural material that comprises much of the mass of plants. Lignocellulose is composed mainly of cellulose, hemicellulose and lignin. Corn stover, Panicum virgatum, Miscanthus grass species, wood chips and the byproducts of lawn and tree maintenance are some of the more popular cellulosic materials for ethanol production. Production of ethanol from lignocellulose has the advantage of abundant and diverse raw material compared to sources such as corn and cane sugars, but requires a greater amount of processing to make the sugar monomers available to the microorganisms typically used to produce ethanol by fermentation. Switchgrass and Miscanthus are the major biomass materials being studied today, due to their high productivity per acre. Cellulose, however, is contained in nearly every natural, free-growing plant, tree, and bush, in meadows, forests, and fields all over the world without agricultural effort or cost needed to make it grow. According to Michael Wang of Argonne National Laboratory, one of the benefits of cellulosic ethanol is it reduces greenhouse gas emissions by 85% over reformulated gasoline. By contrast, starch ethanol, which most frequently uses natural gas to provide energy for the process, may not reduce GHG emissions at all depending on how the starch-based feedstock is produced. According to the National Academy of Sciences in 2011, there is no commercially viable biorefinery in existence to convert lignocellulosic biomass to fuel. Absence of production of cellulosic ethanol in the quantities required by the regulation was the basis of a United States Court of Appeals for the District of Columbia decision announced January 25, 2013. Voiding a requirement imposed on car and truck fuel producers in the United States by the Environmental Protection Agency requiring addition of cellulosic biofuels to their products. These issues, along with many other difficult production challenges, led George Washington University policy researchers to state that in the short term, cellulosic ethanol cannot meet the energy security and environmental goals of a gasoline alternative. History: the French chemist, Henri Braconnot, was the first to discover that cellulose could be hydrolyzed into sugars by treatment with sulfuric acid in 1819. The hydrolyzed sugar could then be processed to form ethanol through fermentation. The first commercialized ethanol production began in Germany in 1898, where acid was used to hydrolyze cellulose. In the United States, the Standard Alcohol Company opened the first cellulosic ethanol production plant in South Carolina in 1910. Later a second plant was opened in Louisiana. However, both plants were closed after WWI due to economic reasons. The first attempt at commercializing a process for ethanol from wood was done in Germany in 1898. It involved the use of dilute acid to hydrolyze the cellulose to glucose, and was able to produce 7.6 liters of ethanol per 100 kg of wood waste per ton. The Germans soon developed an industrial process optimized for yields of around 50 US gallons per ton of biomass. This process soon found its way to the US, culminating in 2 commercial plants operating in the Southeast during WWI. These plants used what was called the American process, a one-stage dilute sulfuric acid hydrolysis. Though the yields were half that of the original German process of ethanol per ton versus 50. The throughput of the American process was much higher. A drop in lumber production forced the plants to close shortly after the end of WWI. In the meantime, a small but steady amount of research on dilute acid hydrolysis continued at the USFS's Forest Products Laboratory. During World War II, the US again turned to cellulosic ethanol, this time for conversion to butadiene to produce synthetic rubber. The Vulcan Copper and Supply Company was contracted to construct and operate a plant to convert sawdust into ethanol. The plant was based on modifications to the original German Skiller process as developed by the Forest Products Laboratory. This plant achieved an ethanol yield of 50 gal per dry ton, but was still not profitable and was closed after the war. With the rapid development of enzyme technologies in the last 2 decades, the acid hydrolysis process has gradually been replaced by enzymatic hydrolyses. Chemical pretreatment of the feedstock is required to prehydrolyze hemicellulose, so it can be more effectively converted into sugars. The dilute acid pretreatment is developed based on the early work on acid hydrolysis of wood at the USFS's Forest Products Laboratory. Recently, the Forest Products Laboratory together with the University of Wisconsin-Madison developed a sulfate pretreatment to overcome the recalcitrance of lignocellulose for robust enzymatic hydrolysis of wood cellulose. U.S. President George W. Bush, in his State of the Union address delivered January 31, 2006, proposed to expand the use of cellulosic ethanol. In his State of the Union address on January 23, 2007, President Bush announced a proposed mandate for 35 billion U.S. gallons of ethanol by 2017. It is widely recognized that the maximum production of ethanol from corn starch is 15 billion U.S. gallons per year, implying a proposed mandate for production of some 20 billion U.S. gallons more per year of cellulosic ethanol by 2017. Bush's proposed plan includes $2 billion funding for cellulosic ethanol plants, with an additional $1.6 billion announced by the USDA on January 27, 2007. In March 2007, the U.S. government awarded $385 million in grants aimed at jump-starting ethanol production from non-traditional sources like wood chips, switchgrass and citrus peels. Half of the 6 projects chosen will use thermochemical methods and half will use cellulosic ethanol methods. The American company Range Fuels announced in July 2007 that it was awarded a construction permit from the state of Georgia to build the first commercial-scale 100 million U.S. gallon per year cellulosic ethanol plant in the U.S. Construction began in November 2007. The Range Fuels plant was built in Soperton, GA, but was shut down in January 2011, without ever having produced any ethanol. It had received a $76 million grant from the U.S. Department of Energy, plus $6 million from the state of Georgia, plus an $80 million loan guaranteed by the U.S. Biorefinery Assistance Program. Production methods. The 2 ways of producing ethanol from cellulose are cellulolysis processes, which consist of hydrolysis on pre-treated lignocellulosic materials, using enzymes to break complex cellulose into simple sugars such as glucose, followed by fermentation and distillation. Gasification that transforms the lignocellulosic raw material into gaseous carbon monoxide and hydrogen. These gases can be converted to ethanol by fermentation or chemical catalysis. As is normal for pure ethanol production, these methods include distillation. Cellulolysis. The stages to produce ethanol using a biological approach are, A. Pretreatment phase, to make the lignocellulosic material such as wood or straw amenable to hydrolysis, cellulose hydrolysis, to break down the molecules into sugars, separation of the sugar solution from the residual materials, notably lignin, microbial fermentation of the sugar solution, distillation to produce roughly 95% pure alcohol, dehydration by molecular sieves to bring the ethanol concentration to over 99.5%. In 2010, a genetically engineered yeast strain was developed to produce its own cellulose-digesting enzymes. Assuming this technology can be scaled to industrial levels, it would eliminate one or more steps of cellulolysis, reducing both the time required and costs of production. Pretreatment. Although lignocellulose is the most abundant plant material resource, its usability is curtailed by its rigid structure. As the result, an effective pretreatment is needed to liberate the cellulose from the lignin seal and its crystalline structure so as to render it accessible for a subsequent hydrolysis step. By far, most pretreatments are done through physical or chemical means. To achieve higher efficiency, both physical and chemical pretreatments are required. Physical pretreatment is often called size reduction to reduce biomass physical size. Chemical pretreatment is to remove chemical barriers so the enzymes can have access to cellulose for microbial destruction. To date, the available pretreatment techniques include acid hydrolysis, steam explosion, ammonia fiber expansion, organozolve, sulfate pretreatment, alkaline wet oxidation and ozone pretreatment. Besides effective cellulose liberation, An ideal pretreatment has to minimize the formation of degradation products because of their inhibitory effects on subsequent hydrolysis and fermentation processes. The presence of inhibitors will not only further complicate the ethanol production but also increase the cost of production due to entailed detoxification steps. Even though pretreatment by acid hydrolysis is probably the oldest and most studied pretreatment technique, it produces several potent inhibitors including furfuryl and hydroxymethylfurfuryl, which are by far regarded as the most toxic inhibitors present in lignocellulosic hydrolysate. Ammonia fiber expansion is a promising pretreatment with no inhibitory effect in resulting hydrolysate. Most pretreatment processes are not effective when applied to feedstocks with high lignin content, such as forest biomass. Organosolv and sporl are the only two processes that can achieve over 90% cellulose conversion for forest biomass, especially those of softwood species. Sporl is the most energy-efficient and robust process for pretreatment of forest biomass with very low production of fermentation inhibitors. Organosolv pulping is particularly effective for hardwoods and offers easy recovery of a hydrophobic lignin product by dilution and precipitation. Cellulolytic processes, the cellulose molecules are composed of long chains of sugar molecules. In the hydrolysis process, these chains are broken down to free the sugar before it is fermented for alcohol production. There are two major cellulose hydrolysis processes, a chemical reaction using acids, or an enzymatic reaction. Chemical hydrolysis, in the traditional methods developed in the 19th century and at the beginning of the 20th century, hydrolysis is performed by attacking the cellulose with an acid. Dilute acid may be used under high heat and high pressure, or more concentrated acid can be used at lower temperatures and atmospheric pressure. A decrystallized cellulosic mixture of acid and sugars reacts in the presence of water to complete individual sugar molecules. The product from this hydrolysis is then neutralized and yeast fermentation is used to produce ethanol. As mentioned, a significant obstacle to the dilute acid process is that the hydrolysis is so harsh that toxic degradation products are produced that can interfere with fermentation. Blue Fire Renewables uses concentrated acid because it does not produce nearly as many fermentation inhibitors, but must be separated from the sugar stream for recycle, simulated moving bed chromatographic separation, for example, to be commercially attractive. Agricultural Research Service scientists found they can access and ferment almost all of the remaining sugars in wheat straw. The sugars are located in the planter unregistered trademark cell walls, which are notoriously difficult to break down. To access these sugars, scientists pre-treated the wheat straw with alkaline peroxide, and then used specialized enzymes to break down the cell walls. This method produced 93 US gallons of ethanol per ton of wheat straw. 1. Enzymatic hydrolysis. Cellulose chains can be broken into glucose molecules by cellulase enzymes. This reaction occurs at body temperature in the stomachs of ruminants such as cattle and sheep, where the enzymes are produced by microbes. This process uses several enzymes at various stages of this conversion. Using a similar enzymatic system, lignocellulosic materials can be enzymatically hydrolyzed at a relatively mild condition, thus enabling effective cellulose breakdown without the formation of byproducts that would otherwise inhibit enzyme activity. All major pretreatment methods, including dilute acid, require an enzymatic hydrolysis step to achieve high sugar yield for ethanol fermentation. Currently, most pretreatment studies have been laboratory-based, but companies are exploring means to transition from the laboratory to pilot, or production scale. Various enzyme companies have also contributed significant technological breakthroughs in cellulosic ethanol through the mass production of enzymes for hydrolysis at competitive prices. The fungus Trichoderma ricei is used by Iogen Corporation to secrete specially engineered enzymes for an enzymatic hydrolysis process. Their raw material has to be pre-treated to make it amenable to hydrolyses. Another Canadian company, Sunopta, uses steam explosion pretreatment, providing its technology to Verenium's facility in Jennings, Louisiana, Abango's facility in Salamanca, Spain, and a China Resources Alcohol Corporation in Soudal. The CRAC production facility uses corn stover as raw material. GenCore and Novozymes have received United States Department of Energy funding for research into reducing the cost of cellulases, key enzymes in the production of cellulosic ethanol by enzymatic hydrolyses. Other enzyme companies, such as Dyadic International, are developing genetically engineered fungi which would produce large volumes of cellulose, xylanase and hemicellulose enzymes, which can be used to convert agricultural residues such as corn stover, distiller grains, wheat straw and sugarcane bagasse and energy crops such as switchgrass into fermentable sugars which may be used to produce cellulosic ethanol. In 2010, BP Biofuels bought out the cellulosic ethanol venture share of Virgatum, which had itself been formed by the merger of Diversa and Cellulose, and with which it jointly owned and operated a 1.4 million US gallon per year demonstration plant in Jennings, Louisiana, and the laboratory facilities and staff in San Diego. CABP Biofuels continues to operate these facilities, and has begun first phases to construct commercial facilities. Ethanol produced in the Jennings facility was shipped to London and blended with gasoline to provide fuel for the Olympics. KL Energy Corporation, formerly KL Process Design Group, began commercial operation of a 1.5 million US gallon per year cellulosic ethanol facility in Upton, Wyoming in the last quarter of 2007. The Western Biomass Energy Facility is currently achieving yields of €45 US gallons per dry ton. It is the first operating commercial cellulosic ethanol facility in the nation. The KL Energy process uses a thermomechanical breakdown and enzymatic conversion process. The primary feedstock is softwood, but lab tests have already proven the KL Energy process on wine pomace, sugarcane bagasse, municipal solid waste, and switchgrass. Microbial fermentation. Traditionally, Baker a unregistered trademark S yeast has long been used in the brewery industry to produce ethanol from hexoses. Due to the complex nature of the carbohydrates present in lignocellulosic biomass, a significant amount of xylose and arabinose is also present in the hydrolysate. For example, in the hydrolysate of corn stover, approximately 30% of the total fermentable sugars is xylose. As a result, the ability of the fermenting microorganisms to use the whole range of sugars available from the hydrolysate is vital to increase the economic competitiveness of cellulosic ethanol and potentially bio-based proteins. In recent years, metabolic engineering for microorganisms used in fuel ethanol production has shown significant progress. Besides Saccharomyces cerevisiae, microorganisms such as Zymomonas mobilis and Escherichia coli have been targeted through metabolic engineering for cellulosic ethanol production. Recently, engineered yeasts have been described efficiently fermenting xylose and arabinose, and even both together. Yeast cells are especially attractive for cellulosic ethanol processes because they have been used in biotechnology for hundreds of years, are tolerant to high ethanol and inhibitor concentrations and can grow at low pH values to reduce bacterial contamination. Combined hydrolysis and fermentation, some species of bacteria have been found capable of direct conversion of a cellulose substrate into ethanol. One example is Clostridium thermocellum, which uses a complex cellulosome to break down cellulose and synthesize ethanol. However, C. thermocellum also produces other products during cellulose metabolism, including acetate and lactate, in addition to ethanol, lowering the efficiency of the process. Some research efforts are directed to optimizing ethanol production by genetically engineering bacteria that focus on the ethanol-producing pathway. Gasification process. The gasification process does not rely on chemical decomposition of the cellulose chain. Instead of breaking the cellulose into sugar molecules, the carbon in the raw material is converted into synthesis gas, using what amounts to partial combustion. The carbon monoxide, carbon dioxide and hydrogen may then be fed into a special kind of fermenter. Instead of sugar fermentation with yeast, this process uses Clostridium legionella bacteria. This microorganism will ingest carbon monoxide, carbon dioxide and hydrogen and produce ethanol and water. The process can thus be broken into 3 steps, Gasification. Complex carbon-based molecules are broken apart to access the carbon as carbon monoxide, carbon dioxide and hydrogen. Fermentation. Convert the carbon monoxide, carbon dioxide and hydrogen into ethanol using the Clostridium legundale organism. Distillation. Ethanol is separated from water. A recent study has found another Clostridium bacterium that seems to be twice as efficient in making ethanol from carbon monoxide as the one mentioned above. Alternatively, the synthesis gas from gasification may be fed to a catalytic reactor where it is used to produce ethanol and other higher alcohols through a thermochemical process. This process can also generate other types of liquid fuels, an alternative concept successfully demonstrated by the Montreal-based company Panicum at their facility in Westbury, Quebec. Hemicellulose to ethanol. Studies are intensively conducted to develop economic methods to convert both cellulose and hemicellulose to ethanol. Fermentation of glucose, the main product of cellulose hydrolysate, to ethanol is an already established and efficient technique. However, conversion of xylose, the pentose sugar of hemicellulose hydrolysate, is a limiting factor, especially in the presence of glucose. Moreover, it cannot be disregarded as hemicellulose will increase the efficiency and cost-effectiveness of cellulosic ethanol production. Sakamoto et al. Show the potential of genetic engineering microbes to express hemicellulase enzymes. The researchers created a recombinant Saccharomyces cerevisiae strain that was able to hydrolyze hemicellulase through co-displaying indoxylanase on its cell surface, assimilate xylose by expression of xylose reductase and xylitol dehydrogenase. The strain was able to convert rice straw hydrolysate to ethanol, which contains hemicellulosic components. Moreover, it was able to produce 2.5x more ethanol than the control strain, showing the highly effectiveness process of cell surface engineering to produce ethanol. Economics. The shift to a renewable fuel resource has been a target for many years now. However, most of its production is with the use of corn ethanol. In the year 2000, there was only 6.2 billion liters produced in the United States, and it has expanded over 800% to 50 billion liters in just a decade. Government pressures to shift to renewable fuel resources has been apparent since the U.S. Environmental Protection Agency has implemented the 2007 Renewable Fuel Standard to use a percentage of renewable fuel in products or face penalties. The shift to cellulosic ethanol production instead of corn has been strongly promoted by the U.S. government. Even with these policies in place and the government attempting to create a market for cellulose ethanol, there was no commercial production of this fuel in 2010 and 2011. The Energy Independence and Security Act originally set goals of 100 million, 250 million and 500 million gallons for the years 2010, 2011 and 2012 respectively. However, as of 2012 it was projected that the production of cellulosic ethanol would be approximately 10.5 million, far from its target. In 2007 alone, the U.S. government provided $1 billion for cellulosic ethanol projects, while China invested $500 million into cellulosic ethanol research. Due to the lack of existing commercialized plant data, it is difficult to determine the exact method of production that will be most commonly employed. Model systems try to compare different technologies' costs, however these models cannot be applied to commercial plant costs. Currently there are many pilot and demonstration facilities open that show cellulosic production on a smaller scale. These main facilities are summarized in the table below. Startup costs for pilot-scale lignocellulosic ethanol plants are high. On February 28, 2007, the U.S. Department of Energy announced $385 million in grant funding to 6 cellulosic ethanol plants. This grant funding accounts for 40% of the investment costs. The remaining 60% comes from the promoters of those facilities. Hence, a total of $1 billion will be invested for approximately 140 million U.S. gallon capacity. This translates into $7 per annual gallon production capacity in capital investment costs for pilot plants. Future capital costs are expected to be lower. Corn to ethanol plants cost roughly $1 3/annual gallon capacity, though the cost of the corn itself is considerably greater than for switchgrass or waste biomass. As of 2007, ethanol is produced mostly from sugars or starches, obtained from fruits and grains. In contrast, cellulosic ethanol is obtained from cellulose, the main component of wood, straw, and much of the structure of plants. Since cellulose cannot be digested by humans, the production of cellulose does not compete with the production of food. Other than conversion of land from food production to cellulose production, the price per ton of the raw material is thus much cheaper than that of grains or fruits. Moreover, since cellulose is the main component of plants, the whole plant can be harvested. This results in much better yields, up to 10 short tons per acre, instead of 4 to 5 short tons per acre for the best crops of grain. The raw material is plentiful. An estimated 323 million tons of cellulose-containing raw materials which could be used to create ethanol are thrown away each year in U.S. alone. This includes 36.8 million dry tons of urban wood wastes, 90.5 million dry tons of primary mill residues, 45 million dry tons of forest residues, and 150.7 million dry tons of corn stover and wheat straw. Transforming them into ethanol using efficient and cost-effective hemicellulose, enzymes or other processes might provide as much as 30% of the current fuel consumption in the United States. Moreover, even land marginal for agriculture could be planted with cellulose-producing crops such as switchgrass, resulting in enough production to substitute for all the current oil imports into the United States. Paper, cardboard, and packaging comprise a substantial part of the solid wastes sent to landfills in the United States each day, 41.26% of all organic municipal solid waste according to California Integrated Waste Management Board's city profiles. These city profiles account for accumulation of 612.3 short tons daily per landfill, where an average population density of 2,413 per square mile persists. All these, except gypsum board, contain cellulose, which is transformable into cellulosic ethanol. This may have additional environmental benefits because decomposition of these products produces methane, a potent greenhouse gas. Reduction of the disposal of solid waste through cellulosic ethanol conversion would reduce solid waste disposal costs by local and state governments. It is estimated that each person in the U.S. throws away 4.4 lb of trash each day, of which 37% contains waste paper, which is largely cellulose. That computes to 244,000 tons per day of discarded waste paper that contains cellulose. The raw material to produce cellulosic ethanol is not only free, it has a negative cost, that is, ethanol producers can get paid to take it away. In June 2006, a U.S. Senate hearing was told the current cost of producing cellulosic ethanol is $2.25 per U.S. gallon, primarily due to the current poor conversion efficiency. At that price, it would cost about $120 to substitute a barrel of oil, taking into account the lower energy content of ethanol. However, the Department of Energy is optimistic and has requested a doubling of research funding. The same Senate hearing was told the research target was to reduce the cost of production to $1.07 per U.S. gallon by 2012. The production of cellulosic ethanol represents not only a step toward true energy diversity for the country, but a very cost-effective alternative to fossil fuels. It is advanced weaponry in the war on oil, said Vinod Khosla, managing partner of Khosla Ventures, who recently told a Reuters Global Biofuels Summit that he could see cellulosic fuel prices sinking to $1 per gallon within 10 years. In September 2010, a report by Bloomberg analyzed the European biomass infrastructure and future refinery development. Estimated prices for a litre of ethanol in August 2010 are €0.51 European euros for 1g and €0.71 for 2g. The report suggested Europe should copy the current U.S. subsidies of up to $50 per dry ton. Recently, on October 25, 2012, BP, one of the leaders in fuel products, announced the cancellation of their proposed $350 million commercial-scale plant. It was estimated that the plant would be producing 36 million gallons a year at its location in Highlands County of Florida. BP has still provided $500 million for biofuel research at the Energy Biosciences Institute. General Motors has also invested into cellulosic companies, more specifically Miscanthus and Coscutter. There are many other companies in construction or heading towards it. Abingo, too, is building a 25 million gallon per year plant in technology platform based on the fungus Miscelia thermophila to convert lignocellulose into fermentable sugars. Poet is also in midst of producing a $200 million, 25 million gallon per year in Emmetsburg, Iowa. Miscanthus, now partnered with Valero, has declared their intention to build a 20 million gallon per year in Kinross, Michigan. China Alcohol Resource Corporation has developed a 6.4 million liter cellulosic ethanol plant under continuous operation. Also, since 2013 the Brazilian company GranBio is working to become a producer of biofuels and biochemicals. The family-held company is commissioning an 82 million liters per year cellulosic ethanol plant in the state of Alagoas, Brazil, which will be the first industrial facility of the group. GranBio's second-generation ethanol facility is integrated to a first-generation ethanol plant operated by Grupo Carlos Lira, uses process technology from Beaker Renewables, enzymes from Novozymes and yeast from DSM. Breaking ground in January 2013, the plant is in final commissioning. According to GranBio annual financial records, the total investment was $208 million. Enzyme cost barrier. Cellulases and hemicellulases used in the production of cellulosic ethanol are more expensive compared to their first-generation counterparts. Enzymes required for maize grain ethanol production cost $2.64–5.28 per cubic meter of ethanol produced. Enzymes for cellulosic ethanol production are projected to cost $79.25, meaning they are 20 to 40 times more expensive. The cost differences are attributed to quantity required. The cellulose family of enzymes have a 1 to 2 orders smaller magnitude of efficiency. Therefore, it requires 40 to 100 times more of the enzyme to be present in its production. For each ton of biomass it requires 15 to 25 kilograms of enzyme. There is also relatively high capital costs associated with the long incubation times for the vessel that perform enzymatic hydrolyses. Altogether, enzymes comprise a significant portion of 20 to 40% for cellulosic ethanol production. Feedstocks. In general there are two types of feedstocks, forest biomass and agricultural biomass. In the U.S., about 1.4 billion dry tons of biomass can be sustainably produced annually. About 370 million tons or 30% are forest biomass. Forest biomass has higher cellulose and lignin content and lower hemicellulose and ash content than agricultural biomass. Because of the difficulties and low ethanol yield in fermenting pretreatment hydrolysate, especially those with very high 5-carbon hemicellulose sugars such as xylose, forest biomass has significant advantages over agricultural biomass. Forest biomass also has high density which significantly reduces transportation cost. It can be harvested year-round which eliminates long-term storage. The close to zero ash content of forest biomass significantly reduces dead load in transportation and processing. To meet the needs for biodiversity, forest biomass will be an important biomass feedstock supply mix in the future bio-based economy. However, forest biomass is much more recalcitrant than agricultural biomass. Recently, the USDA Forest Products Laboratory together with the University of Wisconsin-Madison developed efficient technologies that can overcome the strong recalcitrance of forest biomass, including those of softwood species that have low xylene content. Short rotation intensive culture or tree farming can offer an almost unlimited opportunity for forest biomass production. Wood chips from slashes and treetops and sawdust from sawmills, and waste paper pulp are common forest biomass feedstocks for cellulosic ethanol production. The following are a few examples of agricultural biomass. Switchgrass is a native tallgrass prairie grass. Known for its hardiness and rapid growth, This perennial grows during the warm months to heights of €2–6 feet. Switchgrass can be grown in most parts of the United States, including swamplands, plains, streams, and along the shores and interstate highways. It is self-seeding, resistant to many diseases and pests, and can produce high yields with low applications of fertilizer and other chemicals. It is also tolerant to poor soils, flooding, and drought. Improves soil quality and prevents erosion due to its type of root system. Switchgrass is an approved cover crop for land protected under the Federal Conservation Reserve Program. CRP is a government program that pays producers a fee for not growing crops on land on which crops recently grew. This program reduces soil erosion, enhances water quality, and increases wildlife habitat. CRP land serves as a habitat for upland game, such as pheasants and ducks, and a number of insects. Switchgrass for biofuel production has been considered for use on Conservation Reserve Program land, which could increase ecological sustainability and lower the cost of the CRP program. However, CRP rules would have to be modified to allow this economic use of the CRP land. Miscanthus giganteus is another viable feedstock for cellulosic ethanol production. This species of grass is native to Asia and is the sterile triploid hybrid of Miscanthus sinensis and Miscanthus sacriflorus. It can grow up to 12 feet tall with little water or fertilizer input. Miscanthus is similar to switchgrass with respect to cold and drought tolerance and water use efficiency. Miscanthus is commercially grown in the European Union as a combustible energy source. Corn cobs and corn stover are the most popular agricultural biomass. It has been suggested that kudzu may become a valuable source of biomass. Environmental effects. The environmental impact from the production of fuels is an important factor in determining its feasibility as an alternative to fossil fuels. Over the long run, small differences in production cost, environmental ramifications, and energy output may have large effects. It has been found that cellulosic ethanol can produce a positive net energy output. The reduction in greenhouse gas emissions from corn ethanol and cellulosic ethanol compared with fossil fuels is drastic. Corn ethanol may reduce overall GHG emissions by about 13%, while that figure is around 88% or greater for cellulosic ethanol. As well, cellulosic ethanol can reduce carbon dioxide emissions to nearly zero. Croplands. A major concern for the viability of current alternative fuels is the cropland needed to produce the required materials. For example, the production of corn for corn ethanol fuel competes with cropland that may be used for food growth and other feedstocks. The difference between this and cellulosic ethanol production is that cellulosic material is widely available and is derived from a large resource of things. Some crops used for cellulosic ethanol production include switchgrass, corn stover, and hybrid poplar. These crops are fast-growing and can be grown on many types of land, which makes them more versatile. Cellulosic ethanol can also be made from wood residues, municipal solid waste such as trash or garbage, paper and sewage sludge, cereal straws and grasses. It is particularly the non-edible portions of plant material which are used to make cellulosic ethanol, which also minimizes the potential cost of using food products in production. The effectiveness of growing crops for the purpose of biomass can vary tremendously depending on the geographical location of the plot. For example, factors such as precipitation and sunlight exposure may greatly affect the energy input required to maintain the crops, and therefore affect the overall energy output. A study done over 5 years showed that growing and managing switchgrass exclusively as a biomass energy crop can produce 500% or more renewable energy than is consumed during production. The levels of GHG emissions and carbon dioxide were also drastically decreased from using cellulosic ethanol compared with traditional gasoline. Corn-based versus grass-based. In 2008, there was only a small amount of switchgrass dedicated for ethanol production. In order for it to be grown on a large-scale production, it must compete with existing uses of agricultural land, mainly for the production of crop commodities. Of the United States' 2.26 billion acres of unsubmerged land, 33% are forest land, 26% pastureland and grassland, and 20% cropland. A study done by the U.S. Departments of Energy and Agriculture in 2005 determined whether there were enough available land resources to sustain production of over 1 billion dry tons of biomass annually to replace 30% or more of the national Euro unregistered trademark's current use of liquid transportation fuels. The study found that there could be 1.3 billion dry tons of biomass available for ethanol use, by making little changes in agricultural and forestry practices and meeting the demands for forestry products, food, and fiber. A recent study done by the University of Tennessee reported that as many as 100 million acres of cropland and pasture will need to be allocated to switchgrass production in order to offset petroleum use by 25%. Currently, corn is easier and less expensive to process into ethanol in comparison to cellulosic ethanol. The Department of Energy estimates that it costs about $2.20 per gallon to produce cellulosic ethanol, which is twice as much as ethanol from corn. Enzymes that destroy plant cell wall tissue cost 30 to 50 cents per gallon of ethanol compared to 3 cents per gallon for corn. The Department of Energy hopes to reduce production cost to $1.07 per gallon by 2012 to be effective. However, cellulosic biomass is cheaper to produce than corn, because it requires fewer inputs, such as energy, fertilizer, herbicide, and is accompanied by less soil erosion and improved soil fertility. Additionally, non-fermentable and unconverted solids left after making ethanol can be burned to provide the fuel needed to operate the conversion plant and produce electricity. Energy used to run corn-based ethanol plants is derived from coal and natural gas. The Institute for Local Self-Reliance estimates the cost of cellulosic ethanol from the first generation of commercial plants will be in the $1.90 to $2.25 per gallon range, excluding incentives. This compares to the current cost of $1.20 to $1.50 per gallon for ethanol from corn and the current retail price of over $4 per gallon for regular gasoline. One of the major reasons for increasing the use of biofuels is to reduce greenhouse gas emissions. In comparison to gasoline, ethanol burns cleaner, thus putting less carbon dioxide and overall pollution in the air. Additionally, only low levels of smog are produced from combustion. According to the U.S. Department of Energy, ethanol from cellulose reduces greenhouse gas emission by 86% when compared to gasoline and to corn-based ethanol, which decreases emissions by 52%. Carbon dioxide gas emissions are shown to be 85% lower than those from gasoline. Cellulosic ethanol contributes little to the greenhouse effect and has a 5 times better net energy balance than corn-based ethanol. When used as a fuel, cellulosic ethanol releases less sulfur, carbon monoxide, particulates, and greenhouse gases. Cellulosic ethanol should earn producers carbon reduction credits, higher than those given to producers who grow corn for ethanol, which is about 3 to 20 cents per gallon. It takes 0.76 J of energy from fossil fuels to produce 1 J worth of ethanol from corn. This total includes the use of fossil fuels used for fertilizer, tractor fuel, ethanol plant operation, etc. Research has shown that fossil fuel can produce over 5 times the volume of ethanol from prairie grasses, according to Terry Riley, President of Policy at the Theodore Roosevelt Conservation Partnership. The United States Department of Energy concludes that corn-based ethanol provides 26% more energy than it requires for production, while cellulosic ethanol provides 80% more energy. Cellulosic ethanol yields 80% more energy than is required to grow and convert it. The process of turning corn into ethanol requires about 1,700 times as much water as ethanol produced. Additionally, it leaves 12 times its volume in waste. Grain ethanol uses only the edible portion of the plant. Cellulose is not used for food and can be grown in all parts of the world. The entire plant can be used when producing cellulosic ethanol. Switchgrass yields twice as much ethanol per acre than corn. Therefore, less land is needed for production and thus less habitat fragmentation. Biomass materials require fewer inputs, such as fertilizer, herbicides, and other chemicals that can pose risks to wildlife. Their extensive roots improve soil quality, reduce erosion, and increase nutrient capture. Herbaceous energy crops reduce soil erosion by greater than 90%, when compared to conventional commodity crop production. This can translate into improved water quality for rural communities. Additionally, herbaceous energy crops add organic material to depleted soils and can increase soil carbon, which can have a direct effect on climate change, as soil carbon can absorb carbon dioxide in the air. As compared to commodity crop production, biomass reduces surface runoff and nitrogen transport. Switchgrass provides an environment for diverse wildlife habitation, mainly insects and groundbirds. Conservation Reserve Program land is composed of perennial grasses, which are used for cellulosic ethanol, and may be available for use. For years American farmers have practiced row cropping, with crops such as sorghum and corn. Because of this, much is known about the effect of these practices on wildlife. The most significant effect of increased corn ethanol would be the additional land that would have to be converted to agricultural use and the increased erosion and fertilizer use that goes along with agricultural production. Increasing our ethanol production through the use of corn could produce negative effects on wildlife, the magnitude of which will depend on the scale of production and whether the land used for this increased production was formerly idle, in a natural state, or planted with other row crops. Another consideration is whether to plant a switchgrass monoculture or use a variety of grasses and other vegetation. While a mixture of vegetation types likely would provide better wildlife habitat, the technology has not yet developed to allow the processing of a mixture of different grass species or vegetation types into bioethanol. Of course, cellulosic ethanol production is still in its infancy, and the possibility of using diverse vegetation stands instead of monocultures deserves further exploration as research continues. A study by Nobel Prize winner Paul Crutzen found ethanol produced from corn had a net climate warming effect when compared to oil when the full life cycle assessment properly considers the nitrous oxide emissions that occurred during corn ethanol production. Crutzen found that crops with less nitrogen demand, such as grasses and woody coppice species, have more favorable climate impacts. Cellulosic ethanol commercialization. Cellulosic ethanol commercialization is the process of building an industry out of methods of turning cellulose-containing organic matter into fuel. Companies such as Iogen, Poet, and Abingo are building refineries that can process biomass and turn it into ethanol, while companies such as DuPont, Diversa, Novozymes, and Dyadic are producing enzymes which could enable a cellulosic ethanol future. The shift from food crop feedstocks to waste residues and native grasses offers significant opportunities for a range of players, from farmers to biotechnology firms, and from project developers to investors. The cellulosic ethanol industry developed some new commercial-scale plants in 2008. In the United States, plants totaling 12 million liters per year were operational, and an additional 80 million liters per year of capacity, in 26 new plants, was under construction. In Canada, capacity of 6 million liters per year was operational. In Europe, several plants were operational in Germany, Spain, and Sweden, and capacity of 10 million liters per year was under construction. Italy-based Mossi and Gaisolfi Group broke ground for its 13 mmgy cellulosic ethanol facility in northwestern Italy on April 12, 2011. The project will be the largest cellulosic ethanol project in the world, 10 times larger than any of the currently operating demonstration-scale facilities. See also. References. External links. Genera Energy Incorporated, manager of the largest switchgrass growth program in the U.S. with over 5,000 acres managed over the past 6 years. Ethanol production could reach 90 billion gallons by 2030, backed by Sandia National Laboratories and GM Corporation. Sandia National Laboratories and GM study, PDF format from hitectransportation.org, switchgrass fuel yields bountiful energy. Ethanol from cellulose, a general review. PC Badger, 2002, U.S. DOE Office of Biological and Environmental Research. National Renewable Energy Laboratory, research advances cellulosic ethanol. USDA Forest Products Laboratory, Reuters.com, new biofuels to come from many sources, conference, February 13, 2009 2:50 p.m. Eastern Standard Time, Reuters.com, U.S. weekly ethanol margins rise to above break-even, February 13, 2009 4:01 p.m. Eastern Standard Time, Wired.com, one molecule could cure our addiction to oil, 09.24.07.